Hello everyone, and welcome to another tutorial for BioSE1A. This tutorial is about understanding phosphorylation and protein-protein interaction. You're working with a protein kinase receptor. The receptor belongs to the receptor threonine kinase class because it has 3 threonine residues in the cytoplasmic domain. When the receptor is activated, the 3 threonine residues are phosphorylated, making them negatively charged. This allows the binding to another protein called X. So you are interested in studying the interaction between this receptor kinase and protein X. So you decided to make mutations in the receptor to see how this binding would be affected. Briefly explain what effect, if any, you would expect each of the following mutations to have on the interaction of the receptor and protein X. If you mutate all 3 cytoplasmic threonines to alanine, will protein X still bind? Alanine is a hydrophobic residue. It's not negatively charged, so it can't bind to protein X. Alanine also cannot get phosphorylated. Only serine, threonine, and tyrosine can. So this mutation would prevent binding of the receptor kinase and protein X. Another thing to bring about this receptor is that it has 2 isoleucine residues in the membrane spanning portion. If you mutate these 2 residues to valine, will protein X still bind? Remember that a membrane protein should have hydrophobic residues that span the membrane. Both isoleucine and valine are hydrophobic, so there won't be a dramatic change in the folding of the protein, and the receptor should still be able to localize to the plasma membrane. So most likely, this interaction between protein X and the receptor will still happen. So this mutation may not have any effect on the protein as long as it's still able to fold properly and able to localize to the membrane. Another thing to know about this receptor is that it has 3 phenylalanine residues in the membrane spanning portion. If you mutate these 3 residues to glutamine, will protein X still bind? Most likely, protein X will not be able to bind. Again, remember that a membrane protein should have hydrophobic residues that span the membrane. Now, this mutation changes a hydrophobic amino acid to a hydrophilic amino acid in the membrane spanning region. So this mutation could prevent localization of the receptor to the membrane. If the receptor cannot be present on the cell surface, then it will not be activated. What if you mutate all 3 cytoplasmic threonines to glutamate? Will protein X still bind? Unlike threonine, that needs to be phosphorylated first to be negatively charged, glutamate is already negatively charged. So binding of the receptor to protein X would no longer be dependent on signaling. This interaction could be present all the time. That concludes this tutorial. Thank you for listening, and good luck.